In this video, we're going to talk about B cell activation and how B cells are essential to the acquired immune system response that involves antibodies. Now, we generate B cells and all white blood cells, or blood cells in general, in the bone marrow. The B cells and T cells are derived from lymphoid progenitor cells. Now, we make what's called a naive B cell library in this bone marrow, and these are inactive B cells. Now, there are many millions of groups of B cells, each with a different receptor, this B cell receptor, right? And the idea there is that we have this enormous variety of receptors so that we can recognize an enormous variety of potential foreign antigens. It's essential that we also develop central tolerance, which is removal of B cells with receptors that would bind to our own antigens, to our own cells, causing an activation and an immune system response to our cells, right? It's an autoimmune response, which we don't want. This green little blob up here is our antigen, and that is bound to these very specific B cell group, right? In this case, this one, right? B cells with only this particular receptor that matches that antigen. And upon that matching, we can get activation. Now, we often have T cell help, as we'll see in a moment, but we get a process initiated that's called clonal expansion or clonal selection. And it's really both. We're selecting for a particular B cell receptor. And again, that's really a bound version of the antibody, membrane-bound version of the antibody. And we're going to expand. In other words, we're going to copy this cell. We're going to have this cell divide and divide and divide and divide, right? Multiple generations of mitosis, generating an enormous population of these activated B cells, which really end up largely as plasma cells, this group here. And those are going to be the antibody-producing cells. But a smaller group, a little bit later produced, called memory B cells that are going to be inactive but available for secondary infections. They're actually going to provide protection against this infection in a much more rapid fashion if we get infected later on. Now, the mechanism of T cell help is shown over here in B. And so our naive B cell, right, with our bound version of the antibody, right, this is our B cell receptor, binds our antigen, right, and that initiates phagocytosis, right? So the B cell is a phagocytic cell. We're going to digest that antigen. We're going to then express it on the surface of, or fragments of it, on the surface of the cell using MHC2 proteins, this little guy right here, right? Those MHC2 proteins are presenting the antigen on the surface of the B cell, making the B cell an antigen-presenting cell, for the helper T cells, right? The helper T cells have been parallel stimulated by this same antigen. So this activated helper T cell is expressing a receptor that also matches this antigen. This allows us for a double check, if you will, a backup check, right? The B cell had a receptor that recognized it, but the B cell is going to express it with the MHC2 to the helper T cell to say, hey, do you also recognize this foreign antigen? If it does, then we get this co-stimulatory effect, and we initiate that division and differentiation to produce the plasma cells and the memory B cells. The plasma cells, of course, secreting antibodies by the millions, right, thousands per second, that are going to bind to this foreign antigen, labeling it for destruction.